welcome all of you, and I hope all have done very well during this COVID situation. Uh, After a long time, we are meeting. Today, we have our speaker, that is a dynamic and young Dr. M.D. Devre. He graduated from Mumbai Veterinary College in 1986 and again did masters in 1989, and then he joined the Veterinary College Mumbai as a faculty member, and he remained in the college for nearly 1992 to 2007. During the in service, he was deputed for higher studies for the PhD at Ludhiana. That was the PAU. At present, he is with the Johnson and Johnson. He is the fellow of Johnson and Johnson since 2017, as well as the fellow of our society, that is ISCPT, since 2017. Looking to his professional experience, let me tell that he has put up nearly 20 years of experience in the toxicology for the safety assessment of drugs, cosmetics, and devices in the industry and academic institution. During his working with the Johnson and Johnson since 2009, he joined as a general toxicology manager at the Toxicology Resource Center in 2009. Then 2011, he became associate director of the toxicology. Again, he was escalated to the post of director toxicology in 2014, and again further escalation was there as a senior director and global head toxicology in 2018. Currently, he is responsible for the safety and risk assessment of the cosmetic, over-the-counter drugs, food supplements, and feminine care products for their global launches. He established the Toxicology Resource Center in India to develop safety assessors to assess safety of cosmetic products and ingredients within Johnson and Johnson Consumer Division. He developed over 50 toxicologists in the field of cosmetic safety over last 11 years. Key responsibility he is bearing is to include the product and ingredient safety to support product launches under the NPD, that is the new product development and innovation platforms. He is responsible for supporting to the marketed products, ingredient safety, defense industry, consortia, health authorities globally, addressing any quality issues, post-market safety, and supporting the business models with the third-party manufacturers. He established a third-party support model for the toxicology. He is responsible for the external connect with the industry safety firms in India, within Asia Pacific and globally also. He is responsible for the implementation of the novel technologies, methods like in silico predictions, alternate toxicology models, in vitro models for the risk assessment, representing the J&J in the IPEC, that is Excipient Forum, European Union Consortium, Canada DSR, China FDA Cosmetic Initiatives, CTFAS, and Asian in Asia Pacific. Before that, he was with the Ranbaxy Laboratory in India as a pharma industries from 2007 to 2009. In 2 years, he was associate director in the drug safety evaluation, and the functions were the oversight of preclinical drug development for the new chemical NTG and OTC products for the systemic toxicology, genotoxicity, and safety pharmacology in drug discovery and drug development areas, and support impurity qualifications for the generic compounds assessment. As I told that he also work in the veterinary pharma company, particularly in the poultry industries, for nearly 3 years as a manager. His external engagements are with the industry association within India and within regions, particularly Asians and CTFAS Cosmetic Association, International Excipient Councils. He is the member of Advisory Board of Studies at the Veterinary Universities, establishment of the Toxicology Center at the NIRLH, that is the National Institute of Research in Reproduction and Health, Government of India. He is the editorial board member in various journals. He is reviewer of manuscript for the peer-reviewed scientific journals, invited speaker, lead papers at the national and international conferences. Nearly 11 lectures he has delivered, and conferences he has attended in the tunes of 32, and he has presented papers as much as 30. He has been awarded prestigious the Jawaharlal Nehru Award for his doctorate research work. That was on the Selenium toxicity in 2002. Three times he has been awarded or adjudged the best teacher at the Bombay Veterinary College. He has been awarded best poster presentation as well as award for the standard of leadership at the J&J. He has also received two Orison Awards. To his credit, he has total nearly 80 publications, peer-reviewed journals, that is 27. He has written one book chapter as well as he has published as much as 52 abstracts. So with this biodata and his experience, etc., in the particularly industry, we are happy to have him as a speaker today after a long time. That is the inhalational toxicity and his risk assessment. So Dr. Devra is known to industry as well as to the academics, but for the newer fellows, so and that was his credentials. I also welcome him along with Dr. N. Prakash from the Simoga, Dr. K.K. Sardar, Dr. S.K. Bhavsar, and my executive committee members, particularly Dr. P.K. Verma, Pallavi, and many more. So let us hear Dr. Devre. Whatever experiences he has, he will share with us, and we will be all learning about the inhalation toxicity. Dr. Devre, please.
Thank you, Dr. Thakur. Should I start, Sridhasan, or there are any instructions?
Yes, sir, you can start, sir. Before that, I request all the participants to mute their audio, and also if you have any questions to the speaker, please put it on the chat box. So I will be asking questions to the speaker on behalf of you at the end of the presentation. Thank you. Sir, please, you can share the slides.
Okay. Can you see the slides?
It's coming up, sir. Still yet to come.
Just let me know when you can see the screen.
Yes, sir. Yeah, it's just showing starting to share content. Yeah.
It is showing that Dr. Milind Devre is starting to share the content.
Yes.
We are waiting.
Yeah. Yes, sir. It has come.
It is there.
Okay. Good morning, everyone. Sorry.
Yes, sir. Please go ahead, sir.
Okay. Good morning, everyone, and very happy morning, and a happy New Year, as Dr. Thakur has said. First of all, at the outset, I would like to thank Dr. Thakur, Dr. Srinivasan, Dr. Bhavsar, and the executive committee members of ISVPT for giving me this opportunity and inviting me for this sharing. At the outset, I would really like to acknowledge that I am no expert in inhalation toxicology, because this subject is so vast, it might take an entire career to get expertise in this field. So whatever am I going to do is just sharing what am I knowing till date. Just for the background, what happened maybe a year back or so, I'm the committee member of IPEC, that is International Pharmaceutical Excipients Council. And they are coming with a new guidance, and I was allotted the authorship of intranasal and intraocular and and alternative methods to provide in that. So when I started looking into that intranasal and inhalation kind of drugs or nebulizers, that really created more interest in this topic, and that's what I'm going to share. Is there any? Hello?
There is someone. Susma Garigongar. Please mute your audio.
Yeah, okay.
Yeah, now they can. Yes, sir. Please continue. Sorry, sir. Yeah.
No problem. Yeah, so that was the reason, it created more interest for me. And then during my Ranbaxy days and during Johnson & Johnson days, I've come across few aspects of inhalation toxicology, which I want to share today. So this is what I'm going to do for next 1 hour or so, and would be happy to take any questions after that. And also, if someone else in this audience, I'm sure there are folks who are involved in inhalation toxicity day in, day out, and if they want to add, please do so. Yeah. So what am I going to do is first talk a little bit about I'm going to talk a little bit about the basics. Then we'll go to hazard identification for inhalation, then the exposure assessment, and then the risk assessment and what are the challenges. So because, before talking to the basics also, if you see in general, the toxicities, whatever has happened or would happen is through main 3 routes. One is the oral route, the dermal route, and the inhalation route. Now, oral route, everyone acknowledges and knows that if you ingest something, consider that as the most important one because that is the most common route. Dermal, given the surface area of skin, you are exposed to multiple toxicants day in, day out, and immediate thing what you see is the rashes or allergies or anything which draws your attention. But inhalation,
Switch off your video and continue with audio only, sir. Maybe
okay.
that would help in.
Do that. Am I audible now?
Yeah. Yes, sir. Very well, sir. Yes, sir.
Okay. Okay, so I was talking about the general toxicities. If you compare oral, dermal, and inhalation, oral is very apparent. People are more inclined to know about it and so on. Dermal, given the skin surface area and the rashes and reactions seen immediately, that's the second piece people are worried about. But the inhalation route is such a route that there might be pollutants going in unknowingly to you. They might be causing damage to your respiratory tract and lungs unknowingly to you. So although this is one of the important areas, this often gets neglected. There are a couple of reasons why it so happens, but on both the sides, judging its hazard and then doing the risk assessment has got multiple challenges, and that's the exact reason why I've picked up this topic for discussion today. Okay, if you see the inhalation route, what are the different toxicants you can get? Obviously, first, everything which comes in mind is the environmental pollutants. You always talk of carbon monoxide and lead and so on. So that is known. The second is occupational exposure. In the worker setting, the worker population gets exposed to multiple, inhalants, and that is the second biggest chunk, I can say. The third is the drugs. Obviously it is, but these are intentional, intended. So we know that something is going to go in your respiratory tract. Cosmetics, again, I'll talk a bit more about it. And then there are a few devices. So if you really see, if I talk of drugs, you have got nebulizers, you have got anesthetics, you have got intranasal installations. But as you know, these are prescribed, these are intentional, they are controlled, they are, the doses are controlled, the duration is controlled, and something is going into your respiratory system. Cosmetic is the second one, which is you have got several deodorants, and with the lifestyle changes, more and more people are using deodorants. Then even if it is not very common in India, sunscreens in major part of the world, sunscreens are used. Now, when they are used as spray, maybe 80% goes on skin, but there's 20% which goes in your inhalation tract. Although this is not intended to go into the respiratory tract, it goes. Then there are different aerosols you use day in, day out. Then there are room fresheners you have. There are insect repellents, there are fumigants, and there are biocides. They all are going to go through your inhalation route. The devices, if you go to the devices, obviously there are different devices, but again, the most commons are nicotine inhalers or e-cigarettes, which are most commonly used from the device section. And of course, there are medical devices, which would be again intentional. Now, if you look at all these scenarios and compare the exposure conditions, you can either have a continuous exposure or an intermittent. When we say continuous, there could be ultra short continuous. So suppose if you say your anesthetics, they are ultra short for a short duration given in a continuous basis, and then once anesthetized, it is removed. For short duration, if you say, then your intranasal installations, your nebulizers, your deodorants, they would be short term but continuous. For long duration, of course, you have got these, like you have got mosquito repellents in your house. So all the night through, you are going to get exposed to the fumes for these, and it's a continuous long exposure. Intermittent, obviously, there are multiple intermittent, and your e-cigarettes or nicotine inhalers or your maybe nebulizers, they would be intermittent, but for short duration again. So this would give you the span of different, and of course, the environmental pollutants is again chronic exposure day in, day out on a continuous basis. That's, maybe that would go into the chronic one. So if you go to this first slide, all these would have different types of continuous or intermittent exposures, and you never know how much is going and when it is going. So that is the first complexity. I'm going to talk of complexities one by one, and as they come, I'm going to add to that. So this is the number one complexity. You do not know what is going and when it is going. Okay, then we'll look at the other aspect. What is that which goes to the inhalation route? They are either gaseous or aerosols. Gases, they are two, because these terms are commonly used, so I just wanted to clear first. Gas, definitely it's a pure gas. It's a basic state of matter, like solid liquid gases. Nothing to explain more. It is a single phase gas. At room temperature, it will always be in gaseous phase. Vapors is multiple phase because it's not a state of matter. A liquid is converted into gaseous state. So you will always have a liquid plus gas state, but it is not a basic state of matter. So that is, you'll have these two things. But the inhalation is kind of in gaseous state. Aerosols is tiny particles suspended in air. We define aerosols in different ways, but the basic definition is these are tiny particles, anything suspended in air. Now, it can be dust, it can be fumes, it can be mist. Now, dust is the solid matter. These are large particles in the air, and then they get inhaled. So that something you're getting inhaled. But the thing is that they might be still in the larger. If you see the aerosols, they are less than 1 nanometer to 100 microns. So these dust particles would be near the 100 micron size. The fumes, these are, so small particles, very small particles, which might not be visible, which are in suspended in gases, and then you would see them, they would still go a lower range in the particle size. So soot, Weldon fumes, you would think that you are inhaling some gas, but it is actually the solid particles you are inhaling. The mists are liquids, which are small liquid droplets, which are suspended in gases, and they would be going through inhalation. So when you nebulize the liquids or oil mist, when you use a nebulizer, what you do is you dissolve you have a liquid drug, you put it in a nebulizer and then inhale. What you really inhale is the small liquid particles, which are suspended in gases. So why I wanted to talk about this, because these are the ones which are potential, which have got the potential to enter your inhalation system or respiratory system. And then these are just different types of inhalers. You have got inhaler devices, which is portable versus non-portable. You have got nebulizers and vaporizers. In portable, you have got meter dose inhalers. You have got dry powder inhalers. In meter dose, you have got with pressure, without pressure, breath activated. Like, your e-cigarettes would be breath activated. Pressurized is, your asthma kind of inhalers, metered, which are metered ones, anti-asthma drugs, and so on. So these would be your delivery systems for different inhalers and vaporizers. Again, for vaporizers, you have got different anesthetic vaporizer, electronic cigarette vaporizer. Why I'm just telling this, because you have got different sources where you would get these so-called, pollutants or, unwanted chemicals going into your respiratory system. Okay. The other piece which I want to differentiate is inhalation versus respiratory toxicology. Inhalation toxicology is something which goes through your respiratory system, but through your lungs, it can go into your system also. So it's not only your respiratory system which gets impacted, it is your body getting impacted also. So that is what inhalation toxicology, it's not only restricted to the lungs, whereas respiratory toxicology is only to the respiratory system. So if you see, once you do this inhalation, you will have like systemic effects, and then you will have local lung effects, the respiratory system, and then in between you have got local lung effects also. When I say systemic effects, as I said, something goes to your lungs, gets absorbed in the system, and then you see effects in the other, like you might get reproductive toxicity from inhalation toxicity. Now the only way it can go is from lungs to your system and then to the reproductive system. Locally, just irritation and sensitization to your local respiratory tract or lungs. And then your local responses, you can see in case of these, they are exhibited as maybe acute irritation, asphyxiation, asthma kind of thing, allergies, and they all will be exhibited either as cough or sneezing because the body wants to throw it out. Then if it is a chronic thing, you have got chronic obstructive disorders like COPD. Pneumoconiosis is, you have something which is lodged there, and then finally it's lung cancer, local lung cancer. Okay, with these things, what I want to really talk now is, when we talk of inhalation, it's always related with the particle size. So we always hear particle size, particle size, particle size, and why it is so important. So first thing, your system is designed in such a manner that there will be particles which are respirable, there will be particles which are not respirable, and so on. So as a thumb rule, anything above 10 micron size is not going to go in your lungs or deep lungs or tracheobronchial. They are going to, even though they might be getting into your respiratory system, they are filtered in the nose or oropharynx. They would remain there. They are not going to go beyond, but still you might find them in your nose or or pharyngeal region. Particles between 5 to 10 microns generally will reach your lower, like middle respiratory tract, like here, tracheobronchial region. So, and then particles between 1 to 5 microns are the ones which go to your deep lung and alveoli, which would be launched here. So 10 microns, 5 microns, less than 5 microns. This is clear cutting. And if you even see all the different graphs and everything, what is inhalable will go to your inhalation system. The respirable ones is one which is going to your tracheobronchial and your alveoli. So there is inhalable which goes to your, enters your body. Some enters your thoracic region. Some is respirable to your, deep lungs. But in general, on a conservative side, for all toxicology considerations, what we consider is anything below 10 microns, we consider is going to cause some harm, some effects on your respiratory system, whether it is nose, whether it is pharynx, larynx, whether it is tracheobronchial, or whether it is the deep lungs and alveoli. Right. Now, when we talk of these particles, the basic thing is these particle size are subjected to change over time, I'll talk about more in this. So the first thing is particle settling time. This is what is generally happens. If something is sprayed in air, you will see that 0.5 microns and below would stay in the air before it settles up to 41 hours. 1 micron up to 12 hours, 3 microns up to 1.5. It's 15 hours, not 1.5 hours. And there's some problem, I think. No, this slide is not correct. So 0.5 microns is 41 hours. 1 micron is 12 hours. Yeah. I think these are below these things. I'm sorry for this. And if you are going above your 100 microns, these are nanomicron. These are microns and this is nano. So this will set, the larger the size, it will settle faster, 5.8, right? I'm getting confused now. Okay. The larger the size, it will settle faster in 5.8 seconds. The lower the size, it will take more time. Sorry. So this is clear. 100 microns, 10 microns, 3 microns, 1 micron, 0.5 micron. As the size goes on reducing, you will see the increase in time. So smaller sizes will take more time. Okay. The second aspect is the turbulent versus still air. If your air is still or stagnant, the timing is this. But if it's turbulent, if you are having ventilators, fast fan in your house, this is going to get circulated for more time before it settles. Why I'm talking of this settling time is the more the time it takes to settle, the more is the chance of getting exposed to your respiratory system. That is why lower sizes are more prone to get exposed to your respiratory system. Then most large size would settle fast like this, and they will lose, Small particles would be kind of either they will be surrounding your body or they will lose by exchange with outdoor air. If it is turbulent air, and you have got a good ventilation, they will be thrown out. If it is a closed ventilation, it would still be circulating. And then again, settling is depending on all these other factors also, like impaction. If it impacts something, horizontal versus vertical surface, what happens? There are charge effects. There are positive and negative particles. If they are positive and negative, there is something called as agglomeration, or then there is de-agglomeration. And then even after settling, they might again get released from the surfaces. So the summary is that, each particle size would have a settling time, and that settling time depends on its size as well as your air changes, what it impacts, what charges it have, the release from the surfaces, and whether even if there are few small particles, they can come together So there is a difference between aggregation and agglomeration. Aggregation is, they are fixedly bound. Agglomeration is weak. There's weak bonding, and then they come together. But still, if they come together, they might form large sizes, and it might settle fast, or it is not possible to get inhaled. The other thing is, whenever we talk about inhalation toxicity and particle size, we need to know the particle size distribution. So anything which is sprayed will not be having a constant particle size. Now, this particle size distribution depends on 2 or 3 things. One is the force which it gets emitted. The second is the distance, and the third is the time. Now, and these all dynamics would change. So even if you have this particle size estimation done, it needs to be told, what was the standard. So this we have got from a test which is called as Malvern testing, and they have got a typical thing that they would measure from this much distance and within this time, and with this nozzle. So once you have a distance and time fixed, then the nozzle, or from where it is getting emitted, I'm talking of the drugs and the cosmetics now. From, if you have got aerosol propellant, the nozzle is going to tell about the particle size also, and then you get these type of results out of it. Just for everyone's sake, this tells you that how much is the distribution. So if I want, so when I say now, say D50, that means 50% of the molecules. So what is it now? 50% of the molecules, where is it? Okay, this is between, so you have got here 46, sorry, 50%, right? Where is it? 3%. Okay, here 50% here. This is your 50% of the volume. It is having size 100 microns. So 50% of your molecules are going to be below 100 microns, and 50% are above 100 microns. So if you see DV exact, this is 51%. So at 50%, it is 98 microns. Now, what we are really interested is anything which is below 10 microns. Let us see where it comes. So here, if you see, this is the 10 microns. Now, that means you have got 1% of your particles out of whatever is sprayed. Only 1.4% is below 10 microns. Rest is all is above 10 microns. That means only these many particles, 1.5% particles, are going to get inhaled. Rest, even if you have, say, sprayed, or even if you have sprayed in a day, maybe 20 grams. Out of that 20 grams also, or 20 ml also, only 1.5% is inhalation. This is going to help when we come to exposure and exposure assessment and risk assessment. The other thing about these airborne droplets or particles dynamics is, as I said, timing. So about 35% of airborne droplets, would drop away from your breathing zone in first minute itself. It is going to disperse, 60% in the second minute. So 35% in first minute, 60%. In 2 minutes, you'll lose 60%. In 6 minutes, 90% would get reduced. 95% in 8 minutes, and nothing is detectable after 10 minutes. Even if you spray anything, I'm not talking of drugs now, I'm talking of anything which is intentionally sprayed around your nose or your respiratory system. So whatever gets sprayed, after 10 minutes, nothing is detectable. So this also tells, so the first factor was the particles, the settling time. The second is particle size. The third is timing. So that is why your particle sizes and the deposition depends on source where from it is generated, the force, the distance, and the time. And then the sizes will also depend on how it impacts agglomerates and deagglomerates. These all will come into picture when you do the risk assessment. So just these were some things which I wanted to highlight. Once it enters your body, the other thing is, the solubility comes in picture, water solubility. If it is highly soluble, then it gets settled in your uh, upper upper airways and you can absorb this. So that's why, with higher water solubility, if you talk of ammonia or formaldehyde, they are going to settle in your upper airways. That's why you get nasal cancer and less of lung cancers. If it is low absorbed or less absorption, water solubility, then it goes and settles in your pulmonary region and exert effects in the lungs. And ultra low solubility, if it is more lipid soluble, then obviously, and the small particles would go to your bronchia and alveoli and then get absorbed and get into your circulation. So the first dynamics is on the particle size, the time, the force, and everything, which will tell you how much is going to enter your body. Once it enters your body, then depending on the solubility where it settles, and then again depending on size where it's going to go to the deep lung or not, and then to the circulation. Right. So these were the some basics, I just wanted to highlight before going into the risk assessment piece. Now, in the safety evaluation, these are standard ways. First, you need to know the hazard identification. How much really is the chemical toxic? Then comes the exposure assessment. So in your hazard assessment, you understand the systemic and local toxicity or inherent toxicity. Exposure assessment will talk to you about, how much is really, once you use it, how much is really getting exposed. And the third is then doing the risk assessment based on how much is the exposure and what is the safety limits, and that gives you the risk assessment. So there are 3 steps. We'll talk one by one. So first, let us talk about the hazard identification or hazard characterization. Just give me a minute. Okay. So when we talk about the hazard, definitely this hazard is known by conducting the studies. And you have got in vivo studies, you've got in vitro studies. Now, in vivo studies, when we say inhalation tox studies, they are really, I think, the most complicated ones. What we can know is if you have got oral toxicity studies, you know what is your drug, it is getting dissolved, you analyze it, and then directly gavage it. You know that entire dose is going in the body. Maybe it is rodents or non-rodents. It's very simple. For your dermal toxicity also, you know your vehicle, you dissolve it, you apply whatever rate is there. 1 milligram per centimeter square to 10% of body surface area. So you know how much is applied, how much is on the skin, and how much the animal is getting exposed to. But in case of inhalation toxicity studies, there are many dynamics which come into picture, and I'll talk of that. So in your inhalation tox study, you do have acute toxicity studies, and now recently I've got fixed concentration methods and toxic classic method. There are protocols which takes into considerations the concentration into time. And I can tell you, for inhalation tox, the acute inhalation toxicity is the most common one and most widely used one. There are very, rare cases. I won't say rare, but, the subacute and subchronic studies are conducted mainly for drugs and devices, where for regulatory submissions, and, if something is really getting chronically exposed, but of course, and then as you have NOELs and LOELs in case of oral toxicity studies, you have got no observed effect concentration and low observed effect concentration, which is the endpoint, considering everything. Now comes the complexity. In case of exposures, you either have whole body exposure, nose only exposure, head only exposure, lung only exposure, partial lung exposure. Now, out of these, the whole body and nose only are the most commonly used, and to some cases, the head only. Lung only and partial lung, they are very rarely used. I will talk of these in detail. And then we will talk of the apparatus also. Okay, so these are the apparatus designs. Some examples are there. I don't have all the pictures, but this is the whole body. These are the inhalation chambers wherein the animal is there. The whole animal is kept in this inhalation chamber. This is nose only, where only the nose gets exposed. If you see, the inhalation chamber is here, so you will get exposed here. This is the mask type, nose only for large animals or dogs kind of thing. In addition, talking more about this nose only and whole body exposure. Now, if you see nose only, there are complexities here also. It is either directed flow or a mixed flow. Directed flow is flow past. That means the exhaled air is separated. There's no rebreathing. Now you can imagine, if the same air is, whatever is exhaled by one animal is rebreathed by other, then you have got multiple exposures to that animal, right? And that happens in this mixed flows kind of thing. So if you look at this picture, your input, the inhalation coming is through here. So animal is getting inhaled. The exhalation is going through this. But in this case, this is again going to this second animal. This is again going to the second animal before it goes to the exhaust. That means there is rebreathing. The dose in this case is not very accurately monitored because you do not know how much is inhaled, how much is exhaled, and how much this is rebreathed again. So this is that mixed flow thing. In case of flow past or direct flow, it is a bit opposite. Your inhalation comes from this, your exhalation goes from here. The exhalation is not mixing in that case. So you have, in that flow pass system, you have got a better control on the dosing. That is what I've mentioned here. And this nose only is most commonly used for rodents, as you've seen in the picture. There are a few advantages and disadvantages. Advantages are you use less material, you have got a good control on the exposure. You have got the best control in the flow pass system, a slightly less control on the mixed flow system, right? So you can control the dose. You can study the toxicokinetics in this case, because if you see, the tail of the animal is out and you can withdraw the blood for toxicokinetics. The disadvantage is it takes huge efforts, because each individual animal needs restraint. Usually for acute, you have got 4 hours, and then, exposure, and for chronic, it is still more. So you can imagine the efforts for restraining these animals for continuous exposure. So there is effort there. And then, of course, there is a stress due to this restraint to the animals, which might impact your few biochemical parameters. Coming to whole body system, as I've shown in the picture, you can have horizontal flow or a vertical flow or laminar flow or a turbulent flow. So this meant to maintain the homogeneity. Now, what happens in this case is, for whole animal, it takes some time for equilibration. After the equilibrium, immediately when you put the animal, it is not going to get the same dose. So you have to first saturate or bring that chamber to equilibration, then expose the animal so that it gets a continuous flow of the material or the test item. In this case, the nose only, the equilibration is quite quick, because the area is small. That is one difference between whole body and nose. Now, in case of whole body, multiple animals can be kept in one chamber. You require minimal restraint. So you can use this for chronic studies. But on the negative side or the disadvantage side, when you expose in the whole body in the chamber, you are not only exposing through inhalation route, your entire body is exposed. So your skin gets exposed, something can go through your oral route, and so on. It depends on what material it is. So you really are not characterizing inhalation toxicity in this case. it's very difficult to do a toxicokinetic study in whole body, because once you put it in the chamber for some time, you can only open the chamber and take out the animal for, you cannot have multiple time points for collecting blood samples. So toxicokinetics is very difficult in this whole body thing. You have dose variability. Although there's equilibration, if you have got multiple animals, depending on the feeding rate, there is going to be dose variability. And because of that, you have got poor dose control. So agreed that you cannot have, rodents kept in whole body chambers, and you cannot have, large animals like nose only or something. But, and then yes, so that is why the species also would be deciding what way is to be used. But then these are some of the plus and negative points for these kind of chambers. So that is the first complexity while conducting the inhalation tox study. The second and the most important is the characterizations. How do you have the stable conditions throughout the exposure? It is very, in general, what happens is when you look at the final literature or values, you just get to know the NOEC or no effect concentration or LOAC and LC50 and so on. But you never know what was the way to the study was conducted. It is never, and then you start comparing one LC50 value with other. But then these all factors, or one NOAC value with other NOAC value. But these are all the factors which are going to impact the outcome of your study. If you do not have this monitored clearly, and even if these are monitored, if you change these conditions, your outcome is going to be different. So the first is, how do you characterize the test item? And of course, test item characterization is essential in oral and dermal also, but in this case, it is a bit more challenging. So first, obviously the solubility is going to decide not only the formulation, but where, as I've said in one of my previous slides, the solubility is going to decide whether the compound is going to get lodged in upper respiratory tract or lower respiratory tract. So that is one. The stability over time, obviously, the stability is always there. But it is not only the stability in formulation again, the stability in the chambers, the stability, during the exposure, because if these chambers are at different humidity and temperatures, the stability gets impacted. What we generally do for oral and dermal toxicity is the stability is done at different temperatures. That's fine. And duration. You can have that stability, but once it is getting exposed to these chambers, what happens to humidity? There is a lot of humidity and ammonia built up during the experimentation. Does that impact the stability? Does that impact the solubility? That needs to be studied. And obviously the particle size distribution, which we have talked much about. So that would decide the test item characterization. Now you add the vehicle effects here, and so once you have the vehicle in this for dissolving, it is going to add to the complexity. Does it change the solubility? Yes, of course, it changes the solubility. Does it change the particle size? Does it change the stability? Does it change the reactivity? So although you might have a vehicle control group, what does this vehicle have on test item during exposure? That, unless you characterize that, it's very difficult to compare the outcomes. The exposure chamber, when I say preconditioning, is without the animals also. What are the air changes in the animal? In the chambers. Is there any CO2 saturation? Is there any, what is the oxygen level? Is the animal going to get suffocated? Ammonia building, so on. So these air changes would be definitely important. Second is, how much time does it take for saturation, the chamber equilibrium? If it is taking long time, then you are either wasting the test material or time, or in the initial phase, you're not getting the correct dose. Chamber temperature, as I said, it would have effect on all these parameters plus the stability. And also the comfort of the animal. If the temperature raises, animals are obviously going to be not very comfortable. Then the humidity build up due to respiration. Obviously, Once the humidity builds up, it is going to impact the other conditions. And then the sampling piece. Once, because you're going to monitor the exposure by sampling, so you generate the inhalation or the vapors, it goes to the chamber, and then in the chamber you should have ports to withdraw the sample to really see what is the concentration in air. And in this case, then if these are gas, these are vapors, these are dust, they all will have different different analytical techniques and sampling techniques, and those would be challenging. And of course the characterization when animals are there in the chamber. How fast is the ammonia saturation? What is the effect on animals? What is the effect on stress to the animals? Exposure timing, short term versus long term, intermittent. And then in the chamber itself, is there any conglomeration of the molecules? Once the conglomeration is there, the inhalation, obviously the particle size would increase, there won't be any exposure, and so on. So that we need to see before. So this will all include in the characterization before. So it really takes, so although the inhalation protocol is same, the test item, the vehicle, the format, it's all going to be requiring standardization, and it differs from one molecule to other. Then the other added complexities, whether it is dry powders, whether they are gases, vapors, or whether they are aerosols, like solid aerosols or liquid aerosols. Are there any propellants? Are there any vehicles? These all are going to add to these all factors. And then you will need to really account for these when doing the study. So if you consider oral toxicity and dermal toxicity studies, inhalation toxicity really requires a lot of experience and your expertise. It's not just a kind of technician's job. You really need to know the chemistry. You really need to know the physical chemistry, organic chemistry, and analytical chemistry for for deciding the exposure. So from those studies, you get your NOAC and LOAC values or LC50 values. There are others also, and those values would be used to calculate these many what do you say, comparators or thresholds. For general population, for non-cancer, you've got RFC, that is a reference concentration. Now this reference concentration might be through human studies or from animal studies from NOAC, but then you can use this in your risk characterization. Reference concentration is the concentration to which the population can be exposed lifelong without any adverse effects. Minimum risk level is not for the entire life. It is during the time is specified, maybe short-term duration, long-term duration, work, so that is minimum risk. It is always tied with the timing. Tolerable concentration is, specifically for Canada. And then these are the other, for cancer risk, but I won't go into details because then it becomes a different separate topic for discussion. Similarly, on the worker side, you have got occupational exposure limits, that is OELs. And then you have got the permissible exposure limits and the TL value, again permissible. The TL is threshold limit value, which is again, your what is your exposure limit over the lifetime. But it has got three different things. One is, TWA is time weighted average. So this is for worker population. Time weighted average is for 8 hours in a day. What should be your threshold level where you should not expose? Still is short-term exposure limit over 15 minutes. What should be your limit where above which you cannot go? And ceiling is you should never exceed this concentration. And these are all kind of nobody uses this RML and these values now. Basically what is used is PEL and TLV values. So this is for worker population. Why I am mentioning this? Because many a times when you do the risk assessment, if you don't get your NOAC values or LOAC, the inhalation tox values, then these values could be used as comparators. And sometimes these values are often also derived from your oral toxicity studies by root to root extrapolation. But then it takes a lot of regulatory scrutiny, and once these are decided and human values are there, you can definitely use these values in your risk assessment. And these are the TS systems where you can. So first is definitely the IRIS system, which I've just mentioned. Then there are EPA values which are there, and then otherwise your literature values could be used. These would be the TS system. In vitro toxicity studies, one more thing I just wanted to highlight is on the routes. Before that, you had got nose only and head only and those kind of exposure. I'd also talked about direct lung exposure, but in direct lung exposure you have a better dose control where you expose your lungs to the drug, but then your entire system is not exposed, so you do not know the dynamics of breathing and everything. So there's, although you get to know the effects on the lungs, it is not good from overall respiratory or inhalation toxicity perspective. So this was regarding in vivo studies and the in vivo comparators, including the human comparators. Then there are in vitro studies. Definitely there are multiple in vitro models there, but they would kind of look into the local effects mainly, what happens, and they are more of mechanistic kind of studies. And these would you really go into a different topic of again discussion. What is that? But again, conducting these studies for inhalation toxicity is going to be a bit challenging because you do not know, you're not directly exposing a liquid or a solid to the cells. The cells are exposed through the air or through the vapors, and you really do not know how much is getting exposed. So that again, the exposure thing, how much is getting exposed is a bit of a challenge. And then there are different in vitro models to know the cytotoxicity and other lung effects, including irritation and sensitization. Okay, I'm just looking at that time. Okay, exposure assessment. This is again this, as we said, first is hazard assessment, which we do from these studies which I've just mentioned. Then the exposure assessment becomes the next critical thing. How do we assess the exposure? How much a person is getting exposed. So before going into that risk assessment also, whenever inhalation toxicity comes, a risk assessment comes in picture, please look at these things. First thing is there any significant human exposure by inhalation route? If no, then don't go for it. If chemical is of low volatility and not aerosolized, then you know it is not going to get in the respirable form. If the chemical is too large in particle size, anything about 10 microns or maybe 20 microns, you need not worry also about this case. The same thing here. Aerosol product with more than 99% particles about 10 microns, don't worry. And then chemicals which cannot be generated as gas, vapor, or aerosol, that also you need not worry about, risk assessment. Okay, we've already talked about this. And then, other basic thing is, if it's a propellant, usually what we say is aerosols is inhalable. Everything is an aerosol. Propellant, the ones which are propellant, pressurized propellant, they would generate fine particles. So they are the ones which needs to be really evaluated. The pump sprays, any products with the pump sprays, they will not generate fine particles. They would generate droplets in the range of 70 or 30 to 70 microns. So they are not in the respirable range. Then products with propellant producing foam or soft gels are not suspected to have any inhalable products. And other aerosol, you'll need to really see whether they would generate any particles in that 10 microns range. Then only you need to do a exposure assessment. Then, if you want to talk about exposure assessment, there are three ways of doing it. One is, doing the most conservative way of, calculating what is the exposure, how, and I'll talk about it, how to do. If you do not pass that, if you see that there is still a risk, then you go for actual detecting what is sprayed, how many times it is sprayed, and analyzing it in the atmosphere. And then third is reality-based statistical model or mathematical models is the third tier. But usually we end up in tier 1 and tier 2. And so I'll talk of the tier 1, the most conservative way of looking at the exposure assessment. And this is just a decision tree. So you start having inhalable product. If it is in, I'm now talking of the products. If you have, when you need a risk assessment to be done, or if it is pump spray, you don't have anything, it is about 10 microns, no concern. If it is propellant spray, definitely you have to take it. Now in that propellant spray, you have 85%, which is far off, this which goes far off your face, and 15% is only the one which is near your face, because when you spray something, everything is not on near your respiratory tract. So you only are concerned about this 15%. In that 15% also, the particles, your particle size distribution will decide, whatever is less than 10 microns you take for risk assessment. Anything about 10 microns you don't say. So in the example I gave you, only 1.5% of this is going to go in your respiratory tract. So then you do this. Then the non-propellant fraction is really, the propellants are well characterized, so you need not look at that. The propellant fraction is characterized, so it's not required to look into that. The non-propellant fraction is definitely the one which needs to be looked into. And then you do a kind of exposure assessment, compare this with the NOEL, and do the calculations. In addition to this, you take into consideration the local safety also, and we'll talk about this. So this is, kind of the first year, the calculation by most conservative way, and how it is done. A bit complicated and a busy slide here, but this is what is usually done. So first is, one, you have a product which is sprayed. You have to know what is the external exposure, how much is getting sprayed, and that is calculated by, how much is the amount sprayed. So the dose, how much it is sprayed. For the ingredient, what is the concentration of the ingredient? What is the non-propellant fraction? Because in your propellant sprays, say alcohol or anything, like, it is 85%. Rest 15% is only the ones which have these drugs or the chemicals. So you have the amount, you have the concentration, you have got non-propellant fraction, and airborne fraction is, what is the airborne fraction? Like, when we say airborne fraction, whatever is sprayed, only 20 to 25% is in your respiratory zone. Rest 75 to 80% gets lost, which is away from your breathing zone. So airborne fraction is this, and so by this you know the external exposure. Then comes, as we have said, the particle settling time, the particle size, and everything considering. In first 2 minutes, you're going to have a higher exposure. And then for next 18 minutes, what is going to be the exposure? So you need to calculate exposure in first 2 minutes and then next 18 minutes. After 20 minutes, nothing is there around your nose or your respiratory system. So you need not worry. So you're going to calculate what happens in first 2 minutes, what happens in next 18 minutes. And in that case, what you do is, this is in first 2 minutes, you say IA1, which is this concentration, external concentration, which is how many milligrams per liters you have. And then what is the breathing rate? Breathing rate you have is kind of 15 liters per minute is the human breathing rate. So you know how much liters are getting inhaled in first 2 minutes, and then you know the concentration and then the time. So this is per minute. So this is in 2 minutes how much it is going to get in. Same way you calculate for 18 minutes. In 18 minutes, the breathing rate is around 10 liters, sorry, 10,000 liters. So how much is going to get in next 18 minutes? So that is going to give you exposure of, the EA1 and 2. And then other is, whatever you sprayed, as we said, only 75% is, 25% is airborne fraction, but rest of it is going to land on your skin. So what is going to be your dermal exposure that, or your swallow or from oral exposure, that you take into consideration to calculate the total systemic exposure. I know this is a bit complicated unless you do it multiple times. This is really won't be looked into. Oh, one more thing. When you calculate the inhalation systemic thing, you take into consideration these two multiplied by, default substance exchange factor, means whatever is inhaled, there is partly which is exhaled also. So 25% gets exhaled immediately. So inhalation is considered like whatever is inhaled, out of that only 75% goes or is retained, 25% is immediately thrown out. So that is why you called as a substance exchange rate. And then retention factor or respirable fraction is your particle size, what I said, 1.5%. And then you then divide by body weight. That gives you the systemic inhalation exposure. And then you add what is dermal exposure and whatever is oral exposure to calculate the total systemic exposure. Once you do this, then you go for risk assessment. But this was tier 1. What happens in tier 2? Tier 2 is you if you don't pass in, just give me a minute. In tier 2, you use different models. When you are not able to pass through your most conservative step 1, you go into modeling. And there are various models which are available, the Con Expo and everything. There are different models there. This is one box and two box model there. What is one box model is everything is considered as like you are spraying in a room. Everything is there and there's one inlet and one exposure outlet. So over the time, early exposure, you're going to get exposed to a higher concentration and then a lower concentration. But in practice, it is kind of two box model. That means a box in box, or two box model means, suppose you are spraying a deodorant in your bathroom. So once you spray it, you have got a closed environment, box 1. As soon as you open the door, it gets exhausted in the box, the other, like from your bathroom, it goes into the bigger room, maybe your bedroom or where it gets dispersed. And so, you will get exposure in the next, but the exposure in box 1 is going to be higher and box 2 is going to be lower. There is another way also of saying box 1 and box 2, and that was what we said is what is happening near your face, what is happening apart from, like away from your face. So what happens in first 2 minutes, what happens in next 20 minutes, because after dispersion, what happens. So that is two box model, and there are algorithms there. These are computational models there. They will predict you have to put in how much is sprayed, the particle size, the solubility, and all those parameters, and they will tell you about how much is box 1 and box 2, and that would give you the exposure. This is tier 2. The tier 1 was simple without any modeling. Tier 2 is with these models, and tier 3 is actually maybe you are measuring something and then doing the exposure assessment. Right. And then finally is the risk assessment. You get these values, like from the study, inhalation study, you get the NOAC values, you get the RFC values, the reference concentrations from your regulatory, you get occupational exposure limits or TLV from your occupational exposures. These, and then if nothing is there, then NOAL from your oral, the ADI, TDI, but you will need a root-to-root extrapolation here. If nothing is helpful, then TTC, I'll talk about TTC a bit later. And then once you know the exposure, so your exposure versus your these thresholds, you compare exposure divided by these. If you need to have a margin of safety of 100 for systemic toxicity. For local effects, 25 margin of safety is considered sufficient. Now, having the exposure concentration for local effects, it's a bit challenging. Sometimes you need to do the lung studies, lung exposure studies, because your NOAC would give you systemic and local. So if you really just want to focus on local, it's a bit challenging. But this is what, and as I said, local irritation and respiratory sensitization are more important for the local effects. And finally, TTC, if because at each step, what is the gas, the exchange factor, there are different factors what we mentioned here. So the substance exchange factor, the respirable fraction, the airborne fraction, they all and the non-propellant fraction, they are going to reduce simultaneously the concentration of final what is going into your system. So if the concentration is really minuscule, you can use the TTC levels, the threshold of tox concern. You classify the chemical into these grammar 1 and 3, and then these are the systemic and for inhalation, the systemic effects, and local effect, these are the micrograms. So if your exposure is below these TTC concerns, you need not do anything. Okay, so that is what I wanted to have, as a discussion point. I'm just it's all 12:15. We have got another 15 minutes, but I would like to talk quickly about the challenges. What are there? Obviously, you need a strong analytical support at each stage. Not only formulation characterization, but the characterization of the chamber, the sampling. You require really good analytical support, which is many a times challenging. Then the dosing control. The dosing control depends on the physicochemical characters, calculation of actual dose, considering atmospheric concentration, duration, the other pieces like ammonia, the deposition pattern, etc. And then you require definitely specialized apparatus for that. So that is the second challenge. Then once you do this study, you get the thresholds, but then what are the delivery patterns, exposure patterns? Sometimes it is intermittent, sometimes it is continuous, sometimes short term, sometimes long term, sometimes continuous. And then, as I just mentioned, the exposure assessment is challenging many times. You don't get the actual values. Yeah. So getting the thresholds is one thing. The analytical tool is second thing. Exposure assessment is the fourth piece. And then everything is tied with the organic chemistry and the physical chemistry about the soluble particles, the particle size, the particle dynamics, the impact, and lastly the after doing everything, how it is packaged, the product, because that finally decides again what is going to be the final particle size. You might have studied the actual chemical, but once it goes in the final product along with you might have used the chemical and a separate vehicle, but once it goes to the product, it is a different vehicle, a different particle size which is generated. So how that is going to impact, that is another thing. Obviously nanoparticles is a different game completely. Application of TTC is again questionable, and then last thing is developing in silico models or in vitro models for characterization in local lung effects. That's also a challenge. So these are the challenges, what we face in inhalation toxicity. So as I said in the beginning, inhalation toxicity is really, it's good to discuss, but when it comes to actual practice, it is really, really very challenging. So that's what I had to discuss. Just to summarize, as I said, a very good, interesting topic for study, a very good area to enter to get expertise, but similarly challenging. And if really someone is looking for challenges, this is one area where you can go, and there are very less number of people who would be experts in inhalation toxicity. As I said in the beginning, I'm no expert in inhalation toxicity. This is what I have known during my past experience. But that's all I have. Thank you so much for again to the executive committee members, Dr. Thakur, Dr. Srinivasan, and Dr. Bhausar for giving me this opportunity. Thank you. And I'm happy to take any questions, and please, if there are any additions from the audience, that is also welcome. Thank you.
Okay, sir. Thank you so much, sir, for a very interesting and enlightening uh, lecture. And I believe that you are still holding that teaching gene, though you entered from academics to industry. Really, academics will be missing you, sir. Okay, so there are a few questions in the chat box. Shall I start asking you, sir?
Okay.
Yeah, so there was one question very relevant. It is like in inhalation toxicity studies for drugs, what are the critical factors to consider in study designing to mimic relevant clinical dosing regimen, achieving required human equivalent dose and exposure duration?
So I think this question has two parts. So what was the first part of the question? Can you repeat?
Yeah, in inhalation toxicity studies for drugs, what are the critical factors to consider in study design to mimic relevant clinical dosing regimen?
I would say it is opposite. Once you do this characterization, yeah. So first is when you have what is going to be application, you really need to mimic, like what I said is the intermittent dosing, the dosing pattern. That pattern needs to be mimicked. You cannot mimic the dose. You'll have to get the dosing and then translate into the human equivalent dose and see whether it is safe or not. That. So if I want to say there is going to be mixed, so what needs to be launched, the type of product. So if you have an inhaler, inhalation product or inhaler is there, intermittent dosing, what is going to be my maximum dosing, what is going to be my maximum targeted dose. Now, while designing the study, you'll have to mimic, that the exposure, and if you know the particle size, what is going to be emitted, you will need to design from that, design a target that particle size. Then you'll have to the exposure pattern should be mimicking that way. It's not a very straightforward answer, but you'll have to marry both the things, and then get the answer. I don't know whether I answered it or not, but it is not going to be a straightforward answer.
Yeah, yes, sir. Yeah. Then one question from academic teacher. Like while teaching, we use chalk. Do you come across about inhalation toxicity of that particle?
Yeah, so we will have when you say if you look at the literature, definitely you'll get the values for the chalk particles. But your inhalation is going to be really very low from that place. The particles which are emitted are not in that 10 microns range. That you really have to grind it, and then, of course, I'm not saying there's no exposure, but the exposure is not going to be that huge.
Okay, sir. So, and a few question is like we use isoflurane anesthetic extensively in laboratory animal facilities. Please comment on its toxicity, because a person who are using this isoflurane will be also be exposed to that. So, what are the toxicities due to isoflurane for the like scientists who are exposed to that?
I don't think that you are exposed to isoflurane because it's not an open system where you anesthetize the animal. It is always a closed system. So otherwise, you would always operate in an half-dosed conditions. So I don't think that is possible first. But even if there is some bit of exposure, it is going to be minimal because you have got OEL levels for isoflurane also. So in the manufacturing setup, you have got OEL level for isoflurane, and your exposure is going to be minimal. So, yeah.
Yes. And if a impurity in a drug is respiratory or dermal sensitizer, please comment on its control strategy in drug product or setting the safety limit.
But okay, one question is if your product is a dermal product, or it doesn't matter, your impurity is going to be applied in a manner in which the drug is getting exposed. So if you have a dermal drug, there is going to be impurity which will go through dermal route. If you have got an inhalation product, then definitely you will have to characterize the impurity through inhalation route. And the only way it is done is, what you really do is you take up not a very purified form of your drug during inhalation tox studies. You characterize your test substance in such a manner that you have those impurities already inbuilt during the inhalation tox, so that your impurities also get evaluated during the inhalation tox studies.
Yes, sir. The next question is how we will calculate the settling time in the inhalation chamber during the inhalation exposure in tox studies. How it will be, it will impact on the rate of absorption.
So settling time in the in the chamber itself, it all depends on the sampling. How you sample at different points and then see how much particles you are getting. Now, it is not very easy, and honestly speaking, I don't have a very appropriate answer here because I haven't worked under inhalation, in inhalation lab. But I know in this audience we have got people like Dr. Pore and all. They have been doing that, so maybe they can add on to this. But what I want to just highlight is it's all depends on you just do it the sampling at different time points and then see you have to mimic the exposure scenario and then do the sampling and then analysis to see how much settles in what time. But maybe someone can add also.
Yes, sir. Thank you, sir. And next question is, if a drug formulation, that is diameter 50 is more than 10 micron, is intended for intranasal administration, do we require inhalation toxicity studies for its clinical development?
Yes, because it is inhalation, in your route is in intranasal. You still have to do the inhalation tox study to show that nothing is going to the respiratory tract. Although that 50 microns is not going to go, there are still chances that it is still going to be remaining in your upper respiratory tract and tracheobronchial region. Maybe and that's the way you have to prove its safety. And not just saying that it is more than 50 microns and I won't do it, because your intention is to instill in your nasal cavity.
Okay, sir. And then there are few questions like how to calculate LD50 for in vivo study. Maybe the person is asking about inhalation in LD50.
So you have got different methods now. The guidelines give you good, the OECD guidelines give you good, so it is maybe maximum concentration, or in general it is limit concentrations are there, or you have got, so there are, as I listed down, I can share this. I'm sure you are going to share this text to people, so they will find references to calculate the density.
Sure. Yes, sir. And I think that's all. There was one question, maybe you already answered that you don't have much experience. It said, please share your experience on PK studies after inhalation route of drugs.
Yeah,
so in case you have.
Yeah, not direct experience, but I know the challenges there, and it is possible with rodents, because with the nose only this thing, the toxicokinetic studies can be done in a similar manner as we do for oral studies. But in the whole animal chamber, it is very difficult.
Yes, sir. And I think you already explained about the inhalation toxicology and respiratory toxicology. So the person is again asking how we more differentiate on the terms inhalation toxicology and respiratory toxicology.
So if you get systemic effects, definitely it is inhalation toxicity. Now, many times in regulatory setup, if you have proven that there is systemic exposure, you don't have to do reproductive toxicity through inhalation route. Your oral toxicity and dermal toxicity, oral route toxicity for is more than sufficient, because how much is going into system. Once it goes into system, whether you do characterization through inhalation or other routes, it is same thing.
Yeah. So I think that's all, sir, questions from the chat box. So I may request Professor Silas Bhav, sir, to deliver the vote of thanks.
Thank you, Dr. Srinivasan. I would like to first thank thank Dr. Milind Devre, happens to be a ISPD live member, academician now turned into a technocrat in industry, and he has given a lot of time and the expertise. And the topic was very say complex, but he has explained in a very lucid way, and that was palatable to everyone. So we are on behalf of ISVPT, we are highly thankful to Dr. Devre for sparing his time and sharing the. We are also thankful to Johnson & Johnson for allowing Dr. Devre for this talk. So once again, thank you, Dr. Devre.
Thank you.
I'm also thankful to all the senior pharmacologists and toxicologists who are present the say list and participant list, and they have also served the industry. And I'm also thankful to all the executive committee members who remained present during the say webinar and encourage the other members. I'm also thankful to all the participants from the academia, and many participants I see from the industry also. So those who are interested in the inhalation toxicology, they have attended this webinar. So I'm thankful to all the participants for their active role in this webinar. I'm also thankful to the team of the ISVPT headed by Dr. A.M. Thakur and Dr. Srinivasan and also Dr. Ratnadeep, who are managing this business of the webinar and its pre and post events. So I'm thankful to all of them. I'm also thankful to many say colleagues who have made it possible, and I would like to announce that the webinar talk, today's webinar talk, will be available on the ISCPT YouTube channel. So those who may not be able to remain full-time present today may see the see this presentation on the YouTube channel also. And the last announcement is that Dr. Nitin Bhatia, who is also present today, he will be having a next lecture from his side on most probably on 27th of this month, and he will be on regulatory framework for animal health sector in India. So this is the say tentative topic, and he will be talking on 27th February. So I request all the participants to also take active participation in this forthcoming webinar. So at last, I am thankful to each and everyone, including the speaker, for making this webinar a success. Thank you, everyone.
Thank you, sir. Thank you. Yes, sir. Please.
Thank you, Dr. Devre, for a nice talk and a very interesting topic. Everybody has learned a lot about the inhalation toxicology. Thank you very much. And I thank my all colleagues, particularly Srinivasan, Dr. Bhavsar, Dr. Ratnadeep, and others, and the senior pharmacologists who have attended this webinar. Anything special, Dr. Srinivasan?
Yes, sir.
Thank you, Dr. Thakur, Dr. Srinivasan, and Dr. Bhavsar.
As announced, we'll meet on 27th and listen a very good talk from Dr. Nitin Bhatia from the industry in TAS Pharma, right?
Okay, sir.
Please attend it in large number.
Yes, sir. Thank you so much, sir. Thank you so much to everyone and to the presenter, Dr. Milind Devre. So I request all the participants to fill the feedback form, which is given in the chat box. The link is given in the chat box in order to get a e-certificate for this lecture, okay, this presentation. So kindly fill the feedback form. Thank you, everyone. Sir, should I close the webinar?
Yeah, Dr. Srinivasan, you are permitted to close the webinar. Okay, thanks.
Thank you, everyone.
Thank you.
Thank you, sir.
Bye-bye.
Have a nice day. Have a nice day, all of you, please.
Yeah, bye-bye.